Welcome back to the Deep Dive. Today, we're digging into a group of organisms that, well, they're everywhere, but maybe not top of mind. We're talking phylum Zygomycota.
Right. Often just seen as molds, maybe.
Right.
But there's so much more to them.
Exactly. These aren't just, fuzzy bits on old bread. We're talking fungi that fire spores like cannons, fungi that have these complex chemical signals.
And fungi that are absolutely crucial for our food supply in ways you might not expect.
So our mission today, to unpack why these unsung heroes, and sometimes, let's be honest, villains of the fungal kingdom matter. We're taking a chapter from Introductory Mycology.
And boiling it down, getting to the core insights.
Yep. Think of it as your shortcut to understanding their unique structures, their really surprising ways of reproducing, and their roles, environmentally, in food, even health.
It's custom tailored for you listening to get the key info fast.
Get ready for some aha moments.
What's amazing, I think, is how complex their biology is. You see these things that look simple, but they're shaping ecosystems, impacting us globally.
Let's connect those dots then, from the tiny details to the big picture.
Sounds good.
Okay, let's dive in. Phylum Zygomycota. We're mainly looking at the class Zygomycetes, right? So what makes them? What's the defining thing?
The big one, the defining characteristic, is this special resting spore they make. It's called a zygospore.
A zygospore.
Think of it like a survival pod, really tough, thick-walled, and it develops inside a slightly larger structure, the zygosporangium.
Ah, okay, so the spore is inside the sporangium.
Exactly. That structure, the zygosporangium, forms after two compatible fungal bits called gametangia fused together. And yeah, remembering the spore is inside is key. Older texts sometimes mix that up.
Got it. Zygospore inside the zygosporangium. Survival pod. What about their general build? How do they grow?
Typically, they have what we call a coenocytic mycelium.
Phagocytic.
Yeah, it means they're fungal threads, the hyphae, usually don't have regular cross walls or septa. It's like one long open tube where the cytoplasm and nuclei can flow freely.
Huh. Interesting. So lots of little compartments.
Pretty much. And their walls are made of cool stuff like chitin, chiton, and they're flexible too. Some are dimorphic.
Not even.
They can switch their form, grow as those threads, the mycelium, or as single yeast cells, depending on, the environment. Some even have forms without walls at all, which is pretty unusual for fungi.
Wow. Okay. Adaptable. Where do they fit in the grand scheme of things, evolutionarily speaking? Are they one big happy family?
Well, it's more complex now. Research shows at least 4 main evolutionary lines. It points to them being really ancient with lots of different adaptations.
Like what kind of groups?
You've got the mucorales. Those are often the fast-growing molds we see. Then the entomopterales.
Oh, the insect pathogens? The zombie fungi?
Kind of, yeah. They specialize in infecting insects. Then there are the zoopagales, which are predators actually hunting tiny microscopic animals.
No way. Lethal lollipops, I read.
That's one of them.
That was zero.
Fungus. And then you have the endogonales and glamales, super important groups. Many of these are mycorrhizal.
Ah, the plant partners.
Yeah.
Forming relationships with roots.
Exactly. Forming vital partnerships with the vast majority of plants, helping them get nutrients. Really fundamental stuff.
Okay, so we know who they are structurally, where they fit. Now, the really fascinating bit. How do they reproduce? Let's start with asexual reproduction.
Right. Asexually, they're often incredibly productive. It's mainly through sporangiospores, which, we often just call spores.
Sporangios.
Yeah, imagine this structure, often round, called the sporangium. It sits on a stalk, the sporangiophore.
Right.
And inside that sporangium, thousands of spores can form, maybe 50, maybe up to 100,000. They're made by this cool internal cleavage process.
100,000, wow. And you mentioned the fungus shotgun. That sounds dramatic.
It absolutely is. You see this in Pilobolus. It grows on dung.
Pleasant.
Oh, yeah. But its sporangiophore, the stalk, actually bends towards light. There's a swollen bit under the sporangium that acts like a tiny lens.
A lens made of fungus.
Yep. Focuses light, builds up enormous water pressure, turgor pressure.
Yeah.
And then, whoosh.
Just fires.
The entire sporangium gets launched. Shot off, sometimes up to 2 meters away.
2 meters. That's huge for a fungus.
It is. It's aiming for fresh grass, ideally. So an animal eats the grass, eats the sporangium, and the spores pass through, ready to grow in new dung.
That's incredible engineering.
It even helps disperse other things, like lungworm larvae that hitch a ride.
Wild.
Yeah.
Okay, but is everything that dramatic? Are all their spores launched like that?
Oh, no, not at all. That's Pilobolus being flashy. Many others make smaller structures, sporangiola, with fewer spores, maybe just one sometimes.
Sporangiola,
Yeah.
smaller packages.
Exactly. And there are merosporangious sort of cylindrical spores in a row. Plus, they can make other things like tough resting spores called chlamydospores, or those yeast cells if they're dimorphic.
Lots of options then.
Definitely. And even how the sporangium wall opens up, does it dissolve, break apart? That's also a clue for telling different types apart.
Okay, that asexual stuff is clearly effective for spreading, but what about fungal romance? You mentioned chemistry.
Ugh.
Ah, yes, sexual reproduction. This involves those two gametangia fusing. It's really quite something. It can happen in homothallic species.
Meaning they can mate with themselves. Self-compatible.
Right, or in heterothallic species, where you need two different mating types, like a plus and a minus strain, arbitrarily named.
So how do plus find minus out there in the soil or wherever? Do they send out fungal lonely hearts ads?
Huh.
Yeah.
Something like that, but chemically. It goes back to Blakesley in 1904 noticing incompatibility, and then Burgess in 1924 figuring out it was diffusible chemicals.
Pheromones.
Fungal pheromones. These pheromones are precursors for compounds called trisporic acids. It's really elegant. Each mating type makes a precursor
Okay.
that the other compatible type converts into the active trisporic acid. So trisporic acid only builds up when both compatible types are near each other.
Ah, clever. So the acid is the signal that says compatible partner nearby.
Exactly. And that acid does several things. Ramps up more acid production, tells the fungus to stop making asexual spores for a bit, and crucially, triggers the growth of zygospores. Zygospores. Those are the branches that grow towards each other.
Precisely. Guided by the chemical trail, it's a chemical conversation leading to mating.
So the zygospores meet. Then what?
Their tips swell up, cross walls form, sealing off the gametangia at the very ends. The bits behind become supports, suspensors. Then the wall between the two gametangia dissolves.
And they merge.
Their contents mix, that's plasmogamy. Then their nuclei fuse karyogamy. And that's what leads to the zygosporangium forming, with that single tough zygospore inside.
The survival pod we started with.
Exactly. And those often need time to mature, days, maybe months before they're ready to germinate.
Is this whole sexual cycle common? Do we see it happening often outside the lab?
People used to think it was rare, but we're realizing it's probably more important than we thought. Insects can carry different mating types together.
Oh, accidental matchmakers.
Pretty much. And things like falling temperatures in autumn can trigger it in some species. It suggests it's really key for surviving tough times, for genetic mixing over the long haul.
Okay, so we've got the biology down. Now let's zoom out. Where do these fungi actually bump up against our world? The good, the bad, the tasty.
Right, because they are everywhere. Soil, dung, fruits, flowers, stored grain, you name it. Most are saprobes.
Decomposers. Breaking down dead stuff.
Yep. Nature's recyclers, often called sugar fungi because they grow really fast on simple carbs.
So are they friends or foes, it sounds like?
Both. Definitely both. On the bad side, some are major plant pathogens. Rhizopus stolonifer, that's the common black bread mold, but it also causes soft rot in strawberries, sweet potatoes, big economic losses.
Right. Food spoilage.
And unfortunately, yes, some can infect humans, causing conditions called zygomycosis. Genera like Mucor, Rhizopus, Obsidia, they can cause serious, sometimes fatal infections, especially if someone's immune system is weak.
That's sobering. A serious reminder of their power. But there has to be a good side too, right?
Oh, absolutely. The beneficial side is huge. Many mucorales are industrial workhorses. They make enzymes like amylases, renins, and lots of organic acids. Fumaric acid, which was key in early cortisone production, lactic acid, citric acid, things used in food and industry.
Okay, so industrial chemistry. What about actual food? You mentioned food supply earlier.
Yes. This is really important. Species of Rhizopus and Mucor are used in Asia to ferment rice for making alcoholic drinks, but also, crucially, foods like sufu.
Chinese cheese, right, made from tofu.
Exactly. And tempeh, the Indonesian fermented soybean cake. These fungi don't just preserve or flavor the food, they break down complex molecules, making nutrients more available, boosting the protein quality.
So they actually make the food more nutritious.
Significantly so. These fermented foods are staple protein sources for millions of people. It's a massive contribution to global nutrition, often overlooked.
Wow, okay, that's huge. What about interactions with other organisms, insects, animals?
Oh, they're masters of interaction. We mentioned the entomophthorales, the insect killers. Some are so specific and effective they're used for biological control.
Like controlling pest insects.
Exactly. Entomophaga mymyga helped control devastating gypsy moth outbreaks in North America. a fungal solution to an insect problem.
Cool.
Then the Zooplagales predators trapping rotifers, amoebas. Some are even mycoparasites, feeding on other fungi.
Fungi eating fungi.
Yep. And some use other creatures for transport, like Stylopage, which attaches its spores to mites that live on dung beetles.
Hitchhiking.
It's called forazy. The mite gets a ride on the beetle, the fungus gets a ride on the mite.
Right.
Brilliant dispersal strategy.
Okay, that's clever. And then the really big one, the glomeles.
Ah, yes, the VAM fungi, vesicular arbuscular mycorrhiza. These are maybe the most ecologically important group.
Why is that?
They form partnerships with something like 70% of all plant families, including most of our crops. Their hyphae go into the plant root cells.
Inside the cells.
Yes. They form these intricate structures, arbuscules, like tiny trees where nutrients are exchanged. The plant gives the fungus sugar, the fungus gives the plant phosphate, other minerals, water.
A direct pipeline.
Absolutely. And they also form vesicles, which are like storage sacs. This partnership is critical for plant health, nutrient uptake, especially phosphorus, even disease resistance.
And you can't easily grow these VAM fungi alone in a dish, right?
Generally, no. They really need that plant partner. It shows how deeply interconnected they are with the ecosystem. You have to grow them with a host plant.
It's just such a complex web, which must make classifying them a bit tricky. How do scientists sort out this huge, diverse group?
It is tricky, and the classification has changed a lot over time, especially with molecular data, DNA sequencing, coming in alongside the classic morphology, looking at their structures.
So the family tree keeps getting redrawn.
Pretty much. What used to be broad categories are now split into more distinct families, each with its own set of defining features. It's a much more detailed map now.
Can you give an example or two? What makes a particular family stand out?
Sure, like the Phycomycetaceae, the genus Phycomyces in there, has these amazing metallic green sporangiospores that are incredibly sensitive to light, almost like a simple eye guiding where the spores go.
Wow.
Or the Pilobalaceae, which we talked about, defined by that forceful spore discharge, that cannon mechanism.
Right, the shotgun.
Exactly. Or maybe the Mortioralaceae, some of those smell distinctly like onion or garlic. These unique traits are the clues mycologists use to piece together their relationships.
Okay, what a journey. We've gone from the basics coenocytic hyphae, the defining zygospores inside their zygosporangia,
through to amazing reproduction, that fungus shotgun, the chemical dance of pheromones and trisporic acids guiding mating.
and then seeing their impact everywhere, causing disease, yes, but also
making industrial chemicals, fermenting foods that feed millions, being essential partners for plants, controlling pests. They're woven into everything.
It really shows how these, often invisible fungi are incredibly complex, incredibly adapted, shaping our world constantly.
Absolutely. Masters of adaptation, playing so many different roles.
Makes you wonder, doesn't it? What other hidden biological dramas are playing out all around us, just waiting for us to notice?
Always more to discover.
Well, thank you for joining us on this deep dive into the Zygomyces. We really hope you listening have gained a new appreciation for these amazing organisms.
It was a pleasure trying to connect those dots for you. Remember, the world under our feet and even on our food is teeming with fascinating life.
We look forward to diving deep with you again next time. Until then, keep digging for knowledge.